Back to blood clotting. Yay. Hey, we all need it, right? So a couple people asked me a couple questions about hemophilia and so forth after class, and I just mentioned briefly that there's several places in the scheme where hemophilia can arise, and they arise from deficiencies in any of the clotting proteins, the proteases in the cascade. So they can cause, and there are some that are more common than others, but as you can imagine, if you muck up the cascade, you're going to have some real issues with being able to clot because the signal doesn't make it all the way through. And so that's obviously potentially a real problem. Last time I didn't get a good chance to finish what I was talking about, so I want to come back to that and then move forward. So I talked about briefly how fibrin can polymerize. So when you take fibrinogen and you clip off the A's and the B's, those little short segments off of the fibrin, when you clip these guys off, these little stems that are left are the things that will insert themselves into the little pockets that I showed here on the gamma subunit and also on the beta subunit. And I pointed out how that can make actually a three-dimensional kind of structure if we look at that structure right here. Okay. So we can imagine, here we see the alphas and the gammas linked up, and the betas aren't shown because the betas can insert themselves, as I said, into this dimension coming out. And this makes for a very nice big sort of knot. This gives us, as I said last time, what we describe as a soft clot, and you've seen that as your blood is clotting, when the very first thing that forms to slow everything down in terms of the blood flow coming out of the wound. But to harden that clot, an additional reaction is required, and that reaction creates covalent bonds between the individual fibrins. So as you've seen throughout this course, covalent bonds, of course, are strong. Covalent bonds are difficult to break, and not surprisingly, you're going to have a stronger, firmer structure arising as a result of the linking of individual fibrin subunits by covalent bonds, and that's actually done in a reaction that is catalyzed by an enzyme called transglutaminase. Transglutaminase will join together side chains of glutamines and lysines. Okay? And so wherever these are in relatively close proximity between two different fibrin subunits, this reaction will be catalyzed and result in the formation of this crosslink, which is a covalent bond that helps to stabilize that clot and hold everything together. So that's an important step in that clotting process. Okay. Now, the next thing I want to talk about, I'm going to come back to the pro-clotter and anti-clotters in a second. The next thing I want to talk about is actually another consideration in the clotting process. Okay? And this consideration actually relates to calcium ions. Calcium ions are abundant in our cells, and though I haven't really talked much about them, I will tell you that they are a bit problematic for our cells. And the problem that they pose is that high enough concentrations of calcium, such as if we were to take all the calcium in our cells and let it move around freely, we would have some problems with our chromosomes because the DNA will precipitate. There's enough calcium there to cause the DNA to precipitate. And so calcium in the cells is typically sequestered. We see calcium concentrations rise and fall, but they're usually kept within manageable limits inside of cells so that this precipitation problem doesn't happen. But you can imagine at the site of a wound that we have a ripping open of cells that's occurring, and that ripping open of cells can disrupt the calcium that's found in individual cells. So it wouldn't be surprising to discover that calcium near the site of a wound will be in relatively higher concentration than in places where there's not a wound, obviously. Well, this calcium signal turns out to be important because it's also helping to flag, here's where we need to have a clot. And this flagging turns out to be very important when we consider the enzyme prothrombin. All right? So prothrombin, of course, is the zymogen form of thrombin. And prothrombin has a modification that is done to it that allows it to bind to calcium. That's not part of the cascade. Okay? It's not part of the cascade. That modification can occur and probably is occurring in your blood right now. And it's in preparation for a wound. So that is, if we think about the trigger on a gun, and the trigger on a gun is making sure you're getting ready to fire, okay, what we're doing with the calcium on the site of a wound, what we're doing with prothrombin is preparing it to be ready to be sequestered at the site of the wound very easily and quickly. Well, the way that this works is prothrombin is chemically modified. Okay? It's chemically modified on the side chains of its glutamic acid residues. Okay? So you remember the glutamic acid has a carboxyl side chain. You've seen it as you were calculating charge and all that sort of thing. And the modification that's done to the carboxyl side chain of prothrombin is that an extra carboxyl group is added to it. So it's what we call a carboxylation reaction. And this carboxylation reaction produces glutamic acids now that have two carboxyl groups. This is happening on prothrombin. Okay? Well, the two carboxyl groups turn out to be perfectly positioned to bind to a divalent calcium ion. Perfectly positioned to bind to that calcium that's there. This modification helps prothrombin to really accumulate at the site of a wound because the prothrombin is a perfect binder for calcium at that point. Okay? Now, this reaction that I just described to you is stimulated by vitamin K. Vitamin K is necessary for the carboxylation of the side chains of glutamic acid residues on prothrombin. That allows prothrombin to bind calcium, allows now prothrombin to come to the site of the wound, and again, everything is being directed to very quickly addressing the issue of clotting at the site of the wound. Well, if prothrombin is at the site of the wound, once it gets converted to thrombin, it's going to immediately convert fibrinogen into fibrin. So this localization of prothrombin at the site of the wound is very important. Needless to say, vitamin K is very important. Okay? Now, this turns out to be a factor when we talk about things called blood thinners. Okay? Blood thinners. First of all, vitamin K is a clotting agent. Okay? It will favor clotting. There are competitive inhibitors that mimic vitamin K. Vitamin K is used by the enzyme that makes that carboxylation. There are competitive inhibitors that mimic vitamin K that will stop that carboxylation. Well, if you stop the carboxylation of prothrombin, you're going to have less prothrombin at the site of the wound. You're going to have less tendency to clot. So blood thinning, as it's called, okay, goes by a couple of mechanisms, and the one that we're concerned about here relates to the carboxylation of glutamic acid side chains. These two things here, warfarin and coumarin, are used to block the action of vitamin K, meaning that they block the carboxylation of the side chains of glutamic acid. Okay? They're called blood thinners because the blood literally is thinner in the sense that it's much less prone to form clots. And this is given sometimes to people who have a tendency to clot, or people who suffer from, say, heart problems, which may give rise to clotting situations and so forth. And I'm sure probably many of you know people who take these medications. These medications are pretty critical to get in the right amount. You can imagine that if you gave a person too much blood thinner, they would have no clotting, and no clotting would have some very big consequences, one of which would be that you would be very prone to almost any kind of wound or injury. We think of wound or injuries where we can see blood, but many of our injuries are occurring internally, bruising and so forth, where we never see them. If we were to bruise or do an injury that would cause a bruise where we wouldn't even cut ourselves, we could literally bleed to death internally if we weren't careful with this. This strategy that I've just described to you is actually a critical factor in rat poison. You kill rats by giving them large doses of blood thinner. In fact, we give them warfarin. Okay? The same warfarin that we give to people in low doses to thin their blood. So next time you know somebody who's getting blood thinner, just tell them that they're getting rat poison. They'll be really happy. Okay? All right. So that's what's happening with blood thinners, and they're just blocking the enzyme from binding to and using vitamin K to put that carboxyl group on. Okay, questions about that? Okay. All right. And I've told you how to make clots. It's also important that we think about how to take apart clots. All right? So the body makes clots, but the clots don't stay there forever, because if it did, as we got older and older, we would just become one giant scab, right? The scabs would never go away. They do go away, all right? Some people pick at their scabs and that sort of thing. That's not good. But the body actually has a way of dissolving clots. And the way it dissolves clots is with an enzyme called plasmin. Okay? Plasmin. All right? I don't need that. Plasmin is an enzyme that's a protease, and it will actually attack the fibrin that's in clots, and it will pull it apart, it will tear it apart, and it will, in essence, dissolve the clot. Well, this becomes important as the healing process goes on, because you want to have that clot out of the way so that the normal healing can all occur and so forth. And plasmin catalyzes the breakdown of that clot. Okay? Plasmin, as you might imagine, is something that the body has to be careful of as well, because if we have too much plasmin, we're going to have the same problem that we had with too much blood thinner, right? We're going to be dissolving clots where there really should be some clots, and there's going to be problems associated with that. So plasmin is synthesized also in a zymogen form. It's called plasminogen. So plasminogen, of course, is the inactive form of plasmin. And then the question is, well, how do we activate plasminogen? And we activate plasminogen with an enzyme known as tissue plasminogen activator, or something that you're more likely to call TPA. That's what's on the screen right here. Okay? TPA was the first compound that was actually a first genetically engineered compound that was approved for use in human beings. And it was approved because under some very specific usages, it saved lives and reduced the effects of stroke and heart attack. Show you an example right here. What you see right here is on the left is a heart that has been blocked by a clot. You can see the clot, the blockage right there. The blood flow is sort of shown in white. Okay? That clot was dissolved by the addition of tissue plasminogen activator into the bloodstream directly at this point, which activated plasminogen to become plasmin, and plasmin broke down the clot, and you can see that the blood flow resumed as a result of that. Now, this is, again, something that's used very specifically under very specific circumstances. It's not given routinely because, for the reasons I stated, there could be some problems associated with that. But under very specific circumstances and very specific usages, what we see is that this can actually reduce significantly impacts of heart attack and so forth and stroke in a person who has clotting issues. So TPA is very, very commonly used. I shouldn't say commonly used, but it is used to help treat this type of a problem in people. Questions about that? You guys are a quiet group today? Yes, back there. So if I think I understand the question is, does it break down the clot in a way that the pieces don't later go back and reform a clot? Is that what the gist of what you're asking? Yeah. So obviously it needs to do that. And if you think about it, that clot, after it's had a chance to harden and it's had a chance to form, all those little pockets that those things bind into, they've pretty much all been occupied. So that means then that when you start breaking this thing down, first of all, the pockets are going to be separated so that they're not going to be held together in a mass, so that even if they were to bind an occasional thing here or there, they wouldn't be forming a bigger structure because the bigger structure itself would be gone. So that is a consideration, but the body obviously handles it quite well. Pretty cool result. Okay. Well, that's the last of what I want to say about regulation and control of enzymes. So, yeah, question? Where? Oh, here, yeah, Garrett?
Yeah, so if you have like too much clotting factor, if you have too much of that, what happens?
If you have too much clotting factor, what do you suppose would happen, folks? It'd be the opposite problem. You'd start forming clots in places where you really shouldn't, and that too will be problematic. So one of the things that they do for people, for example, who have heart arrhythmias. Heart arrhythmias where the heart isn't beating in a normal pace. And heart arrhythmias can give rise to internal clots. It's one of the ways that you can get a clot inside of the heart. The first thing that they will do is they will measure the, in essence, level of vitamin K by measuring the tendency or how rapidly clotting will occur in an individual. So they'll take their blood, they'll measure that. And what they do with the blood thinners is they are adjusting the time for clotting. Okay? So a lot of vitamin K means that, wow, this person is really having a tendency to clot. We're going to reduce that, and we're going to reduce it not to the point where they don't clot, but we're going to slow down that process, and in slowing down the process, we're reducing the actual formation of clots. So, Deidre? Yes? Yeah. Yeah. If you mix the blood of hemophilia with somebody who doesn't, the blood of a clotter and the blood of a non-clotter, it will clot, right? Okay. Why does that happen? Why do you suppose that happens? So you're actually replacing the missing component from the person who's missing the clot with the person who has that. So remember that we're talking about a cascade. So if we have one of those enzymes missing in the cascade, we mix it with good blood that has that, the cascade goes on and the clotting will continue. So that's what's happening with that. Yes, right here. Yeah, so there are treatments for people who have blood clotting. Are there treatments for people who have hemophilia? And there certainly are. One of the real wonders of genetic engineering has been the fact that if we know the clotting factor that's missing in a hemophiliac, which we typically will, then you can use a genetically engineered form of that to replace it. And the beauty of that is the genetically engineered form will be a human factor. In the days before we had human factor available, we had to go through thousands of gallons of animal blood to get a factor that was like that, to put into hemophiliacs to replace that factor that was missing. A, that took a lot of blood, and B, that can, in some cases, stimulate an immune response as a foreign protein. So those two things caused real problems for hemophiliacs. Now, that factor is not isolated from blood. That factor is actually grown in a laboratory and then used directly. So that's a real benefit of that. But that's the treatment that's done for hemophilia. Question back there, yes? Yeah, so the question has to do with other types of blood thinners. And she mentioned acetaminophen. Acetaminophen actually isn't, to my knowledge, used at all for blood thinning. At least it wouldn't be at all effective. But aspirin and ibuprofen, two other anti-inflammatories, actually are very useful for that. And that actually comes up, I think we talk about that later this term, with respect to prostaglandins. Prostaglandins are involved in making blood platelets sticky. Blood platelets play a role in the clotting process. And what happens is aspirin and ibuprofen are two different painkillers, they're called NSAIDs, that slow or stop the synthesis of certain prostaglandins. And so by slowing that process, they make the platelets less sticky. And by being less sticky, they have, again, a lower tendency to clot. So that's the other way that we talk about with respect to blood thinners. Two different approaches completely. Okay, yeah, one last question. If you what? So would it be dangerous to use painkillers if you're on blood thinner? The answer is, as always, talk to your doctor. Okay. We turn our attention now to the part of the class that I think almost every student says is probably the easiest part of the whole class. So everybody goes, we can relax, right? Yeah, we can party, we can sit back, we can do nothing for, I don't know, 3 or 4 minutes. Carbohydrates, of course, are critical for us, and they're typically the first thing that we talk about with respect to metabolism. We're getting ready to go into metabolism before too long, because carbohydrates are important energy sources for cells. They are the primary energy that our muscles use. They are the currency that is distributed through our bloodstream from our liver to our muscles as needed, and also stored in our muscles as needed as well, and we'll see some of that. Before I talk too much about carbohydrates, there's a bunch of nomenclature with carbohydrates, and this is why I think students find it's fairly easy, because A, they've been exposed to the nomenclature before, and B, it's pretty much straight memorization. Okay, so carbohydrates are also known as saccharides. Saccharides come from, I think it's Latin, and the Latin name for saccharide means sweet taste. So saccharides are compounds that contain carbohydrates. And carbohydrate, its name comes from the fact that if we look at the formula of these guys, they are hydrates of carbon. What does that mean? Well, if we see, for example, the structure of glucose, we could write the structure of glucose as C6H12O6, which means we could write that as C, all right, parentheses H2O, and put an N by each one, right? So N could be 5, it could be 6, it could be 7, but it's a hydrate, meaning water, H2O, of carbon. That's where the word carbohydrate comes from. Okay, well, when we talk about carbohydrates, we see that they're broken up into several categories. Simple carbohydrates being monosaccharides, meaning that they have one sugar associated with them. Disaccharides, meaning they have 2 sugars. Tri meaning 3, you can kind of get the pattern here. And polysaccharide meaning they have many. If we look at an individual saccharide, we see that an individual saccharide can have 3 carbons, 4 carbons, 5 carbons, 6 carbons, 7 carbons, or 8 carbons. This is an individual monosaccharide. So glucose is a good example. Glucose is a monosaccharide that has 6 carbons. It's a little twisted there, but 6 carbons would be referring to something that's a hexose. So glucose is a hexose. If it has 6 carbons, it is a hexose. If it has 5 carbons, it's a pentose, et cetera, et cetera. Okay? The letters OSE on the end of any biochemical name refers to the fact that we're talking about a saccharide of some sort. Okay? So a triose is a saccharide that has 3 carbons, the tri referring to the 3. Tetrose has 4 carbons, et cetera, et cetera. Okay? All right. When we talk about saccharides and carbohydrates, and I use those terms interchangeably, as you're beginning to see already, we discover that they come in 2 primary chemical forms. You learned about these in organic chemistry. One chemical form being something we call a ketose, and one chemical form we refer to as being an aldose. Okay? So ketoses are saccharides that have a ketone within them, and we can see here on the far left, there is a ketone group right there. This has 1, 2, 3 carbons, so this is a triose. And this triose has a ketone in it. This is actually a compound known as acetaldehyde. I can't even say it. That's not acetaldehyde. That's glyceraldehyde. Well, no, this is glyceraldehyde. This is, God, now I'm drawing a blank. I hate that when that happens in front of an entire class. All right. These are glyceraldehyde. It's not pyruvate. This is, come on, Kevin. Well, it's not going to matter what its name is at the moment because I'm drawing a complete blank. In any event, it's not acetaldehyde. Dihydroxyacetone. Thank you. All right. I was going through the DHAP, which we talk about in glycolysis, and I keep coming up with acetaldehyde. I know it's not acetaldehyde. All right. In any event, the name here isn't that important. The important thing is that I clicked the wrong thing. The important thing is that we have a ketone, and the ketone is shown there. Ketones, you may remember from organic chemistry, are chemically a bit more stable than aldehydes are. That'll become important when we talk about some chemistry later. Aldehydes are fairly reactive. They're fairly easy to oxidize to an acid. And you can see that there's two different forms of this glyceraldehyde, which is what this one's known as. There's two different forms of this glyceraldehyde that's here. And the two different forms, okay, arise from the fact that this carbon right here has 4 different things attached to it. And you remember from your organic chemistry that anything that has 4 different things attached to a carbon creates a molecule that can have those 4 things arranged in 2 different ways. And we refer to those as D and L. And you'll see that because biochemists are lazy, we make this D and L business very simple. Okay? And that's what we have here. But these guys, of course, both being aldehydes. The D and L nature of the compounds are very, very important for us to consider because they really do change the way that the body sees and recognizes these things. Okay? Now, there's a bunch of nomenclature here, and though it looks fairly complicated, I kind of like this figure. So I'm going to sort of step you through what all of these individual things mean. Okay? So I'm going to start here with the term stereoisomer. Okay, I won't use constitutional isomer. I don't use that term, but a stereoisomer. When I talk about a stereoisomer, I talk about 2 things that differ in their stereo configuration, meaning the way that those 4 things are arranged around a carbon. Okay? So stereoisomers in saccharides, as we will see, can exist in multiple forms. Right? So, for example, if we have 2 things that are stereoisomers of each other, and those 2 things are mirror images of each other, we'll see that there's some that are not mirror images, and they're mirror images, we refer to them as enantiomers. So a really good enantiomer, you've already seen it. There's one form of glyceraldehyde known as the D form, and another form of glyceraldehyde known as the L form. If I told you that I had D-glucose, and I said, what is the enantiomer of D-glucose? It turns out to be very simple. You would say L-glucose. Okay? It's possible because there are multiple asymmetric centers in most saccharides. It's possible that we can have 2 compounds that have asymmetric centers, but that are not mirror images of each other. Here's a good example. Okay? These are both aldoses. These are both hexoses, so we could call them aldohexoses. Right? And when we look at the orientation of these hydroxyl groups with respect to each other, of course, left and right side makes a very big difference in terms of the stereochemistry, we discover that glucose looks like this guy on the right, and altrose looks like the guy on the left, and we could not put a mirror here that would reflect this one onto that one. They're not mirror images. So if we have 2 different stereoisomers that are the same size and the same chemical type, meaning they both have 6 carbons and they're both aldehydes, if they're not mirror images, we refer to them as diastereomers. So in order for something to be diastereomers, they have to have more than one asymmetric center. Okay? And if we break down the diastereomers even farther, we can look at some other considerations. Okay? If we say that we have 2 diastereomers that differ in the configuration of only 1 carbon, we have epimers. The only difference between mannose and glucose is the configuration of that carbon right there. Every other carbon has the same configuration. So these two guys are epimers. Now, I haven't talked yet about cyclization of sugars, but we will talk in a minute about the fact that straight-chain sugars, if they're 5 or 6 carbons long, can fairly readily form ring structures. And when they form ring structures, they create a new asymmetric carbon. That new asymmetric carbon is called the anomeric carbon. And the anomeric carbon is carbon number 1 if it was an aldose, and carbon number 2 if it was a ketose. So here's carbon number 1, and we see a new asymmetric carbon that's created. It's drawn in the down configuration for glucose, for alpha D-glucose, and it's drawn in the up configuration for beta D-glucose. These are both forms of glucose, but they differ in the position of the anomeric carbon. When that happens, we call them anomers. Okay. So we have stereoisomers, we have enantiomers, we have diastereomers, we have epimers, and we have anomers. Okay? Anomers will always have the same chemical name. I will have alpha D-glucose, beta D-glucose, alpha D-galactose, beta D-galactose. Those will always be anomers because we're talking about a backbone that consists of the right sugar differing only in this. Alpha, as we will see, indicates the hydroxyl is down on the anomeric carbon, and beta indicates the hydroxyl is up on the anomeric carbon. 2 questions back in the back.
Is that supposed to be O6 at the bottom of altrose, or is it an extra bottom?
Say what?
There's 7 oxygens in that of altrose under diastereomers, and it says O6 at the bottom. So is that a typo at what point?
Oh, actually, look at there. There's an extra. Somebody's put an extra hydroxyl on there. Yeah, good eyes. Yeah, that's the problem. Yeah. So in the case of this guy right here, this would have an H. Yeah, good eyes. I was author of a textbook one time, and the textbook was in the 3rd edition by the time I got to it. So we're looking, as you guys, I'm sure you know, looking at textbooks, they don't change figures much from one textbook to the next and the next edition. And it was the 3rd edition of this textbook that was used everywhere. And I looked in there, and I looked at one of the figures that was in it, and I said, that carbon's got 5 bonds. And nobody had noticed it. It goes through all kinds of review and everything, and everybody everywhere, and nobody had noticed it. So I felt very proud about that. So you should feel proud, is what I'm telling you. Good for you. Okay, other questions? Was that your question also, or comment? Good eyes, yeah. Okay, so there's a bunch of nomenclature there. I think you should know what those different nomenclatures refer to. Okay. This figure shows what we call the straight chain form of different monosaccharides. And several of these are structures I think you should memorize. Now, I keep memorization to a minimum. And the only reason I make anybody memorize some of these structures is because some of these structures appear everywhere. You're going to need to learn them for another class anyway, so we might as well learn them right here. The structures from here that you need to memorize are ribose, glucose, okay, galactose, and fructose. If you look at these, you'll see that they're not that different from each other. Let's look at ribose. Ribose is a pentose, meaning it has 5 carbons. It's an aldose, meaning it has an aldehyde. And its structure is pretty straightforward, folks. It's got 3 hydroxyls all on the right side. Ribose is the sugar that we find in RNA. A related structure found in DNA is known as deoxyribose, and it contributes the D part of the DNA. Glucose. If you memorize the structure of glucose, you get the others pretty easily. When I was in college, I had to memorize the structure of glucose, and I remember glucose was right, left, right, right. Right, left, right, right. Okay? Whoa. Okay. Galactose is just like glucose, except for it differs in the configuration at this carbon right here. So instead of right, left, right, right, it's right, left, left, right. Fructose is just like glucose, except for it's a ketose instead of an aldose. So, if we number the carbons, 1, 2, 3, we start with number 3, we see that it's exactly the same configuration as glucose. Simply, it's a ketose instead of an aldose. Now, the last thing I'll tell you about the sugars relates to the D and L nature of those sugars. Okay? They say biochemists are lazy people. Right? They give names to things instead of going through and designating all the individual configurations of each carbon. So, for example, galactose differs from mannose because it has different asymmetric carbons. Okay? If I were to say, what is the structure of L-glucose, I would hope that what you would draw me would be the mirror image of glucose. Now, what's the L sugars look like? Well, the L sugar configuration of glucose, instead of being right, left, right, will be left, right, left, left. If I were to draw a sugar on an exam, and I say, is this a D sugar or an L sugar? The way that you would tell is by looking at the next to last carbon. The configuration of the next to last carbon will tell you if it's a D sugar or it's an L sugar. This is a D sugar because the hydroxyl is drawn on the right. If I move this hydroxyl to the left, I create an L sugar, but if that's the only thing I moved, it's not L-glucose, because L-glucose is the mirror image of that. It would mean all those would have to be flipped. Understand? Okay, so know the difference between D and L, and know the difference between D and L of a given sugar. The sugars that I've just designated to you, ribose, glucose, galactose, and fructose, all exist in ring structures, and yes, you should know the ring structures as well, and as you will see, they're pretty straightforward. Well, ring structures brings me to my next topic, which is some nomenclature relevant to the rings. I think in organic chemistry you've seen how these ring structures can form. I'll show you a figure in a second. But before I do that, I want to make you familiar with nomenclature that we use for these. Okay? This compound that you see at the top is called pyrene, and you'll see that it has 6 members to its ring, one of which is an oxygen. When we see glucose form a 6-member ring, it'll form something that looks like that, and we call it, therefore, a pyranose, P-Y-R-A-N-O-S-E, again referring to the fact that the OSE says that it is a sugar. Something that has 5-membered ring, and notice that's not 6 carbons, that's a 6-membered ring, but one of those is oxygen. If I said glucose forms a pyranose, you have to say, where's the other carbon at, right? It's not a part of that ring. It's sticking off of the ring somewhere. Typically, it's sticking off over here. We'll see that in a minute. Okay? Ketose, like fructose, that has 6 carbons, will commonly form something called a furan, or in this case, we refer to it as a furanose, a 5-membered ring. And we remember that fructose has 6 carbons, okay? And because it has 6 carbons, there must be 2 carbons hanging off the ring there somewhere, and we'll see how that goes as well. Okay. Now, glucose can form 5-membered rings, not as commonly, and fructose can form 6-membered rings, not as commonly, and I'm not going to make you know the alternate forms of those, so I'll show you which forms I want you to know. Okay. This shows the cyclization of glucose to form a pyranose. All right? So here's the straight chain form, and the straight chain form of glucose we refer to as the Fischer structure. F-I-S-C-H-E-R. The Fischer structure. The ring structures are what we call Haworth structures. H-A-W-O-R-T-H. In solution, this ring structure can form very readily. And not only can it form very readily, but it can go back and forth between one form and the other. You notice the back and forth arrows going there. Okay? Back and forth between one to another. So it actually can go from here, back to here, up to here. That's what happens. Okay? Well, this formation of the ring, as I noted earlier, creates a new asymmetric carbon. This carbon number 1, which looked like that over there, the carbon number 1 over here only had 3 things attached to it. It was not an asymmetric carbon. Over here, that carbon now has 4 things attached to it. It has an oxygen, it has a carbon, it has a hydrogen, and it has a hydroxyl. This new asymmetric carbon, as I noted before, we call the anomeric carbon. And the orientation of the hydroxyl relevant to that anomeric carbon is designated as alpha if the hydroxyl is in the down position, and beta if the hydroxyl is drawn in the up position. Okay? Give you a trick question for the exam. Trick question for the exam says, what's the alpha or beta configuration of D-glucose in the Fischer chain, in the Fischer structure? And there is none, because alpha and beta requires an asymmetric carbon, and in the straight chain there is not an asymmetric carbon at carbon number 1, right? Yes? How do you tell the difference? Alpha has the hydroxyl down, and beta has the hydroxyl up on the anomeric carbon. Okay. Let's see. What else can I say there? This shows how fructose forms what commonly, the most common structure it forms is the 5-membered ring. Okay? So for fructose and for ribose, they both commonly form 5-member rings. You should know the 5-member ring structure of those two. For glucose and for galactose, they most commonly form 6-member rings, and you should know the 6-member ring structures for those. Well, here's how fructose forms, and you'll notice that when this anomeric carbon is formed, the anomeric carbon is always the one that was the carbonyl. Notice here's carbon number 2, and carbon number 2 is now shown over here. Because it's carbon number 2, we see carbon number 1 sticking off the ring, and we see carbon number 6 sticking off the ring, just like we saw carbon number 6 sticking off the ring in glucose. The difference here is that glucose had the carbonyl on carbon number 1, which meant it became part of the ring. In the case of fructose, carbon number 1 is not making the ring, and so it sticks off of it, as you see here. All right, so if I look at this, this is alpha D, and I don't care if you call it fructofuranose or whatever, okay? I might say, what are all the different things we could call it? You could say, well, it's a furanose, it is a ketose, it is a hexose, right? But if we look at this, the alpha designates the position of the hydroxyl. So if I say alpha, the hydroxyl is down, which means that the CH2OH, carbon number 1, has to be up. And conversely, if I have beta, then the hydroxyl would be up and the CH2OH would be down. Okay. There's a whole bunch of stuff you don't have to memorize. Okay. You can now see the alpha. Well, actually, the top 2, I guess you should know. Alpha is down, beta is up. But you don't have to worry about fructose in the 6-membered ring. That's good news. And there's common structures that you see in 6-membered rings: ribose, glucose, fructose, and galactose. And I'm not making you learn mannose, so you're spared there. You don't need to know deoxy-D-ribose either. Okay. Now, the next thing I want to talk about are conformations. Okay. Good job. We've mentioned before, or I've mentioned before, that we have to think about, in proteins, for example, the issues of steric hindrance, because folding can bring things close together, but there are going to be limits how close they can get together because of considerations of steric hindrance. Turns out steric hindrance is a factor in monosaccharides as well. And that factor arises because there are interactions that can happen between the 2 A's that you see right here. The 2 A's, this is a CH2OH, and this is an OH that's here, and they're relatively bulky groups. And these groups can be oriented in a couple of different ways. We refer to them as boat and chair. And now I'm not talking about different chemical forms of the sugars. I'm talking about the same sugar bent in different ways. So this could be glucose, for example. in what we call the boat form. You notice that this is in the beta configuration. This is hydroxyl here. Okay? And we see it in the chair form over here. All right? But in this case, it's still up. It's just now pointing outwards instead of pointing upwards. The same sugar. This is called the chair form because if we draw it out, it looks kind of like a chaise lounge. There's the back of the chair. Well, you got to use your imagination. Back of the chair. There's where your butt goes, and there's where your feet go down there. Okay? The boat form looks like sides of a boat, like that. Well, it turns out that the boat form, as you can sort of see with your eyes, is more sterically hindered than is the chair form. And consequently, when we analyze the structures of these in solution, we find that both forms actually can occur, but that the chair form is preferred. And it's preferred because there's less interaction, less steric hindrance that's occurring in the chair form. Okay. I think it's time for a song. Oh, the limerick! Oh my God, we didn't do the limerick of the day. Sorry, guys. Let's get the limerick of the day. This is yesterday's or last time's limerick. This limerick should be different, and this limerick is: some cheaters at golf often say dropping scores from their card is okay. These devious folks reducing their strokes see no need to play the fair way. Okay, golfers, all right. Here's a song that somebody else is going to sing. It's called Thank Goodness My Blood is Clotting. It's recorded by a couple people I know that did a great job on this, and if I can get the sound. Here we go. 'Cause I cut myself back. All right. See you guys next time.